Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasis from Department of Biotechnology and Bioinformatics from JP University of Information Technology, Solan, Himachal Pradesh. Today we are going to talk on module Introduction to Mass Spectrometry, which is coming from paper Biophysical Technique 1. There are following learning objectives of this module. First is the introduction and basic principle of mass spectrometry and its major components, which involves method of sample ionization, types of mass analyzers, and separation of sample ions. Lastly, detection and recording of sample ions. It is also covered application of mass spectrometry as a learning objective. So before going to start, I want to ask one question. What is mass spectrometry? Actually, this mass spectrometry is an analytical technique that can identify a substance, its chemical nature and quantity present in a sample by measuring its mass to charge ratio. Mass spectrometry can identify compound on the basis of the atomic sample composition of the molecules and their charge state. By mass spectrometry, blind analysis of unknown sample is possible since mass spectrometry does not require comprehensive prior information of the sample. Okay, broadly these techniques involve several points. Number one, that creating gas phase ions from analyte molecule or the molecules. Then second one is separating ions according to their mass to charge ratio, that are m by z, and lastly measuring the abundances of these ions. Broad applications of mass spectrometry. Actually, mass spectrometry can be applied for qualitative and quantitative analysis. It can provide information about mass of the atoms and the molecules which are present in a substance. It can also tell you the molecular structure determination, whether it's organic or the inorganic. And routinely or nowadays you will find the identification of characterization of biomaterials or biomolecules are also associated with mass spectrometry. And these techniques involve the proteomic analysis, which will be covered in different sections. The mass spectrometer. Mass spectrometer is an instrument which converts the molecule into ions so that they can be moved about and manipulated by external electric and magnetic fields. The mass spectrometry consists of following three fundamental components. Number one, ion source or ionizer. Second is mass analyzer, and third system is detector system. Number one, ion source. An ion source is the first component of mass spectrometer that creates gaseous ions from the substance being studied. A small sample is ionized generally to cations by the loss of an electron. The two most common ion sources devices are matrix assisted laser desorption ionization, or we can say it's MALDI, and second is Electrospray ionization. The second component of mass spectrometer is mass analyzer. A mass analyzer is the second component of the mass spectrometer that receives ionized masses and resolve them on the basis of charge to mass ratios and output them to the detector. The most common mass analyzers are time of flight, quadrupole mass analyzers, and quadrupole ion trap mass analyzers. The third system or the last component of mass spectrometer is detector system, which is required for detecting the ions and recording the relative abundances of each of the resolved ionic species. The results are displayed on a chart as mass spectrum. Now, what is mass spectrum? A mass spectrum is a plot of the ion signals as a function of the mass to charge ratio. The spectra are used to determine the elemental or isotopic signature of a sample, the masses of a particle and of molecules, and to illustrate the chemical structures of molecules such as a peptide or other chemical compounds. Mass spectrometer. This figure shows the basic working of a mass spectrometer. You will find the samples are firstly ionized with the help of MALDI or ASI. Then these samples were further proceed towards mass analyzer where they are segregated on the basis of their mass and charges and relative abundances. And lastly, they will be detected with the help of electron multiplier. Basic principle behind mass spectrometry. For mass spectrometry analysis of a compound, the sample has to be introduced into the ionization source of the instrument, and production of gas phase ions of the compound is done by electron ionization. For this, the sample passes through an electron beam which knocks off some electrons from the molecule and turns them into ions. A mass spectrometer generates multiple ions from the single sample. Ionic forms of molecule of samples are advantageous as ions are easy to manipulate than neutral molecules. These molecular ions undergo further fragmentation. These ions are separated according to mass by charge ratio, that is M by Z, in the analyzer region. The resulted mass spectrum is displayed in the form of a plot of ion abundances versus mass to charge ratio. Thus, the structure and nature of precursor molecule is elucidated. The separated ions are detected, and this single signal sent to the data system where M by Z ratios are stored together with the relative abundances for presentation in the format of any M by Z spectrum. Now, for mass spectrometry, first thing is how to introduce a sample into mass spectrometer. Pure sample can be inserted directly into the ionization source. That can be done. But what about the complex mixture? Complex mixture are first processed through various chromatographic techniques like HPLC, gas chromatography, or capillary electrophoresis. After separation by these technologies, a series of components which can be then finally entered into mass spectrometer sequentially for individual analysis. Now, after sample introduction, the another thing comes, that's the sample ionization. Ionizer is a component which is required for sample ionization. There are several methods of sample ionization used in mass spectrometry. Electron impact, fast atom bombardment are the older methods, while MALDI, ESI are the newer one. In biological systems, you will find the use of matrix assisted laser desorption ionization or electrospray ionization are routinely utilized, and you will find these are the method of choice for conducting biological samples identification. Both positively charged and negatively charged sample ions are generated to the proton affinity of the sample. The ionization method to be used depends on the nature of the sample and type of the mass spectrometry. Now coming to the newer question, that is, what is the mode of ionization, whether it is positive or the negative? The answer comes from two terms. One is for proteins, another is for DNA. For example, if a sample has a functional group that is readily accept protons, such as amide or amino group found in peptides and proteins, then positive ion detection is utilized, for example in proteins. While if a sample has functional groups that are readily lose a proton, such as carboxylic acid and hydroxyls found in a nucleic acid and sugar, then negative ion detection is utilized, and it is utilized for DNA as well as sugars. Methods of sample ionization. This figure shows the list and specification of different ionization methods. Among these, electron impact and fast atom bombardment are older methods and usually required in specific needs. For example, electron impact is utilized for environmental work using GCMS. The most common ionization methods for biological systems are ESI and MALDI, as well as atmospheric pressure chemical ionization. Both positively and negatively charged sample ions are generated to the proton affinity of the sample. The ionization method to be used depends on the nature of the sample and type of the mass spectrometer. For example, if you see electron impact ionization, this method is mostly used for GCMS and is considered as hard ionization technique because it causes the fragmentation of ions. Ionization of gaseous and volatile substance can be made by energetic electrons. In this method, a beam of electron is formed by heating a filament bias at a negative voltage compared to the source. The electrons are further used for bombardment to the gaseous phase molecules, and because of this, the molecular ions are produced. While in case of fast atom bombardment, this technique is involved for ionization of non-volatile compounds. In fast atom bombardment, sample is mixed with a matrix like glycerol, a mixture of dithiatriol and other compounds to keep a sample in a liquid state as it enters at high vacuum ion source. The matrix also protects an analyte from being damaged by the high energy bombardment particles. Ionization is done by bombarding a sample with a beam of atoms, typically xenon, accelerated at the voltage of 8 to 10 kilovolt energy. The ions formed by fast atom bombardment are adducts to the molecule, where adducts could be proton, sodium, potassium, or ammonium. The other techniques like ESI and MALDI are discussed in further slides. Matrix assisted laser desorption ionization, that is MALDI. MALDI is a soft ionization technique where the sample is embedded in a solid matrix, for example 2,5-dihydroxybenzoic acid, which absorbs energy at the wavelength of laser 337 nanometer and transfers a proton to the sample. Laser causes the desorption and ionization of the matrix and analyte either by cationation or by deprotonation. Molecular ions are then accelerated into the MS analyzer. MALDI mostly forms singly charged ions and preferentially coupled with time of flight mass analyzers. Shows MALDI ionization, in which absorption of UV radiation by chromophoric matrix and ionization of this matrix. In the second figure, you will find dissociation of a matrix phase change to a supercompressed gas, charge transfers to analyte molecule. In the third figure, you will find expansion of matrix at supersonic velocity and light trapped in the expanding matrix plum, that we call explosion or popping. Now coming to the advantages of MALDI, or why MALDI is utilized for majority of the proteomic experiments which are generally happens in day-to-day life or for biological identification of proteins. Firstly, that this system is totally automated system. Second thing is, sample can often be used directly without purification after in-gel digestion. That makes life very easy. Third thing is, it is very good for sequence genomes and it will rapidly gives you data after identification. Moreover, unlike ESI, MALDI generates spectra that have just a singly charged ion form, and it is easier to conclude the identification from this MALDI experiment. It generally happens with the positive modes as M plus H plus ions. Generally, This is more robust machine as compared to ESI, and because of this it tolerates salt and non-volatile components. And if this happens in ESI, you will never get the results. Moreover, it is easier to use and maintain, and it is capable for higher throughput, and you will get approximately more than 100 proteins in a single day because of this MALDI experiment. Electrospray ionization or ESI. Currently, electrospray ionization is one of the most popular ionization techniques and generally employed in the mass analysis of polar molecules. ESI has an advantage in its easy compatible with LC. In ESI, the sample is dissolved in a polar volatile solvent and introduced by a narrow capillary. Just see in the left panel of the figure. The electrospray is produced by applying a high voltage, approximately 3 to 4 kilovolt, on a flow of liquid at atmospheric pressure. The concurrent flow of nebulizing gas, that is generally nitrogen, is used to direct the spray emerging from the capillary towards the mass spectrometer. The spray enters into the vacuum system and the droplets are desolvated by heat, vacuum, and high voltage. Further, the ions are ejected from the droplets and pushed into the mass analyzers. Just see the right panel of the figure. Ions may contain multiple charges, allowing the detection of very large molecule on analyzers. Usually, formic acid is added for protonation of the sample molecule in a positive ionization mode, while ammonia solution or a volatile amine may be added for deprotonation of sample in negative modes. Peptides and proteins are usually analyzed in positive mode, whereas nucleic acid and saccharides are investigated in negative ionization condition. Advantages of electrospray ionization. Electrospray ionization has several advantages. Firstly, electrospray ionization can be easily interfaced to LC. Secondly, absolute signal from electrospray are more easily reproduced, therefore better quantification. Mass accuracy is considered better as compared to other ionization. There are multiple charges which are produced by electrospray ionization as compared to the MALDI. These electrospray ionization can be modified to nanospray system with a flow rate of less than 1 ml per minute. This technique is very sensitive and requires less than a picomole of the material. Lastly, it has some disadvantages also. One of the major disadvantages is that this electrospray ionization is strongly affected by salt and detergents. Now, coming to the next section, that is section 2 of a mass spectrometer. Generally, people are using 3 types of mass analyzers which may be utilized as a single mass analyzer, or sometimes they can be coupled together. The 3 analyzers are time of flight mass spectrometer, quadrupole mass spectrometer, and quadrupole ion traps. Time of flight mass spectrometers. This is one of the simplest mass analyzers. It measures the M by Z ratio of an ion by determining the time required for it to transfer the length of a flight tube. The time of a flight of an ion is proportional to the square root of the mass by charge ratio given a constant accelerating voltage, which is given by the formula time of flight equal to constant M by Z ratios in a square root. It also includes an ion mirror at the end of the flight tube which reflects ion back through the flight tube to a detector and serves to increase the flight tube length. Ion mirrors are also corrects the small energy difference among ions. Both these factors contribute to an increase in a mass resolution. Figure 3 shows the representation of a MALDI along with the TOF analyzer. Slide number 18 shows the combination of MALDI ionization source and time of flight mass analyzer, in which you will find the source is getting energy from UV of 337 nanometer and the samples are irradiated in the form of positive ions, which will be followed for analysis in time of flight mass analyzer and lastly detected on microchannel plate detectors. Mass spectrometry equation for time of flight mass analyzer: M by Z is equal to 2 voltage multiplied by time of travel in a square root divided by length of the drift tube. Quadrupole mass analyzer. As the name suggests, this analyzer consists of 4 circular parallel metal rods and electric field between them is quadrupolar. Just see in the figure. An electric field is generated by applying a radio frequency which is 120 degree out of phase to the opposite pair of rods. Thus, the shape of one waveform is equal and opposite to the other. Direct current of equal magnitude but opposite polarities with respect to the ground is supplied to each pair of opposing rod. The electric field created in the mass analyzer acts as a filter in that it only allows ions of certain masses, basically the resonant ions, to pass down the gap between the 4 rods, and therefore the quadrupole acts as a mass to charge filter. A narrow band filter is produced due to the relation of the radio frequencies to DC voltage. This band filter allows the ions of one M by Z value to take a stable trajectory between the alternating rods to the detector, while ions of different M by Z non-resonant ions collide with the rods and become neutralized. Quadrupole ion trap mass analyzers. This analyzer also works on the same principle as the quadrupole analyzers mentioned before. It uses an electric field for the separation of the ions. The analyzer is made up of a ring electrode with grounded end cap with a specific voltage. Please see the figure given in the slide. The ions enter into the cavity between the electrode through one of the end caps, and there the electric field causes the ions of a certain M by Z value to orbit in the space. On increasing the radio frequency voltage, heavier mass ion orbits become more stabilized, while light mass ion become less stabilized and collide with the wall and therefore do not detected by the detector. An increasing mass selective ejection of trapped ions occur in ion trap mass analyzers by gradually increasing the applied radio frequencies. In contrast to quadrupole mass analyzers, ion trap mass analyzers allow ions to be stored and then selectively ejected from the ion trap and increased sensitivity. Detectors of mass spectrometer. It is the last section of a mass spectrometer. Earlier, detectors used photographic films, but nowadays, detectors or the ion channels and electron multipliers produce electronic signals via second-degree electronic emission when struck by an ion. Timing mechanism integrate these signals with scanning voltage to allow the instrument to report which M by jet has struck the detector. These mass detectors need to constant and regular calibration. It is showing an electron multiplier that we call diode. These detectors monitor ion current, amplifies it, and signal is then transmitted to the data system where it is recorded in the form of mass spectrometer, which is the most common data representation. The M by jet values of the ions are plotted against their intensities to show the number of components in the sample, the molecular mass of each component, and the relative abundances of the various components in the sample. The type of detector is supplied to suit the type of analyzer. The most common are the photomultiplier, the electron multiplier, and the microchannel plate detectors. Now coming to the application of mass spectrometers. Mass spectrometry has a wide number of applications in current scenario. Use of mass spectrometers is not limited to the routine industrial application but also required academia for research purposes as well as for medical uses. Among these, the following applications are being utilized at higher level. Number 1. Characterization and quantification of biomolecules. Sample confirmation to determine the purity of the sample, to verify amino acid substitution, to detect post-translation modification, to calculate the number of disulfide bridges. All these things happen for characterization of proteins, and this study we call proteomics. For further, you will find that the amino acid sequencing or the peptide mass fingerprinting, sequence confirmation, de novo characterization of peptide, identification of peptides or their macromolecular structures can be done with the help of mass spectrometry. Similarly, one can use the oligonucleotide sequencing with the help of mass spectrometry or the characterization of quality control of oligonucleotide. The next application of mass spectrometry is related to enzyme technology, that enzyme identification and their subunit modification or the protein digestion can be seen or can be analyzed with the help of mass spectrometry, and this is related to reaction monitoring of enzyme technology. Mass spectrometer can also be utilized for medicinal chemistry, pharmacokinetics, and drug metabolic processes, and definitely it can be utilized for pharmaceutical industry. Likewise, you will find this mass spectrometry has a very big role in clinical diagnostic and prognostic services. For example, screening of specific disease marker or the biomarker can be done with the help of mass spectrometry, drug testing can be done, and several other things. Likewise, the environmental analysis of polyaromatic hydrocarbon analysis or water quality or food toxicology can be done with the help of mass spectrometry. Likewise, geological like oil composition can be seen with the help of mass spectrometry analysis. So students, let's now summarize what we have learned in this current module. Number 1. What is mass spectrometry? Mass spectrometry is an analytical technique used to identify the type and amounts of chemicals present in a sample by measuring their mass to charge ratios and abundances of their gas phase ions. Second is mass spectrometer, which is an instrument. Mass spectrometer is an instrument capable of producing binome of ions from sample under investigation, separating these ions according to their mass by charge ratio, recording the relative abundances of the separated ion species as a mass spectrum. The mass spectrometer consists of three fundamental components, and you will find these three fundamental components in each mass spectrometer. Number 1, ion source. Number 2, mass analyzer. And number 3, detectors. Ion source and mass analyzers are generally operated in vacuums, and these are also under the influence of electric and magnetic components. Okay, now coming to the ionization source. Ionization source are basically required for ionizing the molecules to be identified. Commonly used ionization sources are electron impact ionization, fast atom bombardment, electrospray ionization, matrix assisted laser desorption ionization, and atmospheric pressure chemical ionization. In these techniques, you will find ESI and MALDI are generally utilized for biological sample identification, and these are soft ionization techniques. Now, mass analyzer is the second section in a mass spectrometer which receives ionized masses, and these ionized masses are finally resolved on the basis of their charge to mass ratios. Examples of mass analyzers are time of flight mass analyzer, that's we call TOF, quadrupole mass analyzers, and ion trap mass analyzers. After that, the ion current corresponding to the different species are amplified, and these are either displayed on a oscilloscope or a chart recorder, or they are stored in a computer, and this section is required for as a detector. Now coming to the application part. Mass spectrometers serve for the establishment of the molecular weight of the molecule, structure of both inorganic and organic compounds, and the identification and determination of analyte in a complex mixture. This also helps or establishment of different proteomics studies which is happening these days in a biological sections. Thank you very much.